Thank you very much for being here. We don't often, for our Education Excellence campaigns, have a formal kickoff in this way. And so this was a bit of a risk for us to see if people, particularly on a second night after parent conferences, would have people come. But we thought this was such an exciting topic that we may entice some people to hear more about what we're doing. And I'm just so thrilled about this topic. So I figured I would tell you a little bit about why we came up with this topic, but I wanted to first start by just sharing a personal story, which is something I usually don't do, but I think there's a connection. I grew up in a small town outside of Boston, Melrose, Massachusetts, and then, in those days at least, it was much more of a middle-class town. And in my science experience, I loved science. I loved my science classes, but I never actually was a big Michael Crichton fan, which thrilled me even more as a high school boy interested in science. But I never really saw the path for career in science. I was one of the few African American kids in my school, and I was a high achieving student. And where I saw the path was in history, actually. I wanted to become a history teacher. And it was actually conversations with my teacher, projects and research that I did within those classes that sort of helped me to see that pathway. In science, I remember sort of my culminating honors Bio 2 course. I remember doing a project on the company Merck. That was like the only pathway that I saw of biofield. And my wife, who also grew up in rural Pennsylvania, and she loved science through middle school until she had some teachers who she felt like completely moved her away from that career, did not think that was a career for her. And so when I came to Lexington High School as a history teacher, when I was 22, completely different type of community than what my wife grew up in, what I grew up in. I was completely blown away by the number of students who would tell me, when you'd ask, what do you want to do in your life, who wanted to be doctors. And I was just, I think for a couple of years, I just was baffled. I would say to my wife, so many kids at the school want to be doctors. It's just totally amazing. How is this possible? And what I realized is that all of them, or almost all of them, were children of doctors. And so when you think about as science has become more and more prominent in terms of career opportunities, a lot of the different, as more pathways open up, we still find that the people who are going into those jobs tend to be male, tend to be white, tend to be more affluent. And we still see the gaps that we see in high schools in terms of achievement, or in terms of who is taking our science courses. We see those same gaps in the career fields as well. And many of you who are in those fields probably could agree to that. The reason why I came into education is to address those kinds of gaps. And the reason why I came to Newton North High School is because this is a school that I think more than any, at least in this area, and I think nationally, who care more about giving opportunities to all students. And so that's why I think this is an exciting opportunity. Why this particular is that I was listening to NPR one day driving home and was listening about the CRISPR machine. And as someone who spent a lot more time thinking about history and being a principal than science, I had not heard much about genetic editing, and so I went to Heather Haines, our department chair, and said, oh, this CRISPR machine sounds really cool. And she's like, what the heck is my principal talking about? CRISPR machine? And I just kept talking on and on and on, and she's just sort of giving me this look that she rarely gives me, but just sometimes gives me this look of just like, where is he going with this? And I think one of the moments that I just thought was really neat was that it solved a problem that I constantly struggle with, which is baldness. Is that they talked about the ability to possibly cure baldness, which I thought was pretty
Awesome.
Do you remember this conversation?
Yeah.
So obviously, as we learn more and more about, as I've learned more and more about genetic editing, and just in the news just a couple of weeks ago with this doctor in China who it just leads to significant ethical questions. It just is so exciting that we are a school that's willing to kind of think about how we can give an opportunity for all of our students to be able to learn about this type of science, this type of technology. That's pretty amazing. And the fact that we have educators who can hear their crazy principal sort of talk about stuff that he doesn't really know what he's talking about, but get excited about it, and they can actually know what to do with it and make it happen is totally amazing. And what I'd say is that in having worked in many schools now is that we have a science department here who believes in that equity value of our school. And so what's truly amazing is they're willing to take a risk in taking this on. And so I want to just thank you for being open to this idea as well. And I hope tonight that you're going to hear what this type of science is. Some of you, this may seem something that you have a lot of knowledge about, and I'm sure you'll have a lot of great questions. And for some of you, you may be like me, that you just are an avid NPR listener and get your 5 minutes of information and do whatever you want with it. But I do believe that what this will do for us as a school is that it will open up a lot more eyes to the sciences. It will energize more students about the sciences. And those children who were sort of in the same boat that my wife and I were in, who just need to sort of find that passion and see that pathway, are going to have an opportunity to do that, regardless of whether they choose that field or not. So with that, I believe our PTSO is going to give us a little bit of information about the process, and then our scientists will actually talk about what they're doing. Thank you very much.
Thanks, my name's Lori Knowles, and together with Melissa Condoridis, we are the co-chairs of the Educational Excellence Fundraising Campaign this year. And you may already know that the Educational Excellence Fundraising Campaign is a campaign. It is the only annual school fundraiser that we have for Newton North, and we fundraise for the priorities and needs that the school asks us to fundraise for. So this is their choice, and then they ask for PTSO to go to the parent and guardian community and to raise money for their chosen need and their project. And you probably already received the materials that we mailed out just prior to, or I guess actually just after Thanksgiving you would have received them. If you accidentally recycled them without an opportunity to look at them, we have more up here, or if you think there's somebody that you know who'd be interested in this, by all means come and take some more information. Melissa and I had the opportunity to meet with Heather to learn. We needed to learn before we could write the materials what we were actually raising money for. And so we sat down with her and we learned about the experiments, and we saw some of the machinery and got to hold it and touch it. And we thought, this is so interesting and exciting to actually see it and understand what the students are learning, because sometimes high school education is a bit of a black box, unlike elementary school education. And so when Elizabeth came to us, where's Elizabeth at the back, and was talking about an event, we thought this would be terrifically exciting to allow people to see and learn what's actually happening in the classroom, what our kids are going to be learning. And I can tell you, as I work in STEM, I'm actually a bioethicist, and I have 2 children, both of whom are humanities kids. So for me, I have a senior who's in forensics, and I have a sophomore who is going to be taking biology, like anybody who's got a sophomore or a junior this year, or a freshman, they will be taking biology. And for me, I feel it's even more important for my humanities kids who might go on and do humanities, and I think they will, to see that what I think of as the beautiful mystery of plant, animal, and human DNA, to be able to see it and get really close and get engaged in it, regardless of whether they go on and do something professional with it or not. I think it creates opportunities for them to see and understand in a fuller way their relationships to other people and their relationships to the natural world. So that's why we're really excited about it. And the campaign's kicked off already. And we have some really exciting news, which is we have a generous family that has donated $15,000 to match $15,000 of donations. So the more donations we get, that match will actually match each of those donations. So that's really exciting because the goal this year is $43,000. That's for equipment that's been priced out, and that's bigger than anything we've done in recent history. So we're excited with the opportunity to actually meet that goal and get the equipment into the classroom this year. So if you have a junior, we're talking about then being able to use that equipment this year. We were just talking about purchase orders, and as soon as we get the money, we're starting to purchase equipment to get it in the classrooms. So, and if you have any questions, Melissa and I are going to stay through the program, and we look forward to answering your questions and hopefully to your support for this campaign. Thank you so much. Heather.
Hi, so I'm Heather Haines. I'm the Science and Tech Engineering Department Chair here at Newton North. This is my third year here at the school, and I'm just so thrilled to be here on behalf of the entire biology team, 10 teachers, and the whole science department of 30, asking for your support as we move forward in creating a bigger, better program for all of our students. So when I came to North 3 years ago, one of the first things I did was interview everyone in the department and ask, what are you interested in? What do you need? What is it that you're working on right now? And what I kept hearing from the biology team was, we really want to do more biotechnology. We know that we're not doing enough for our students. They live in this changing world. And what was funny is they would all reference CRISPR as well. So when Henry was with me in this meeting, I have a very, here's the opposite side of the story here. I'm thinking to myself, I have to figure out how we can get more funding for the biology team. And then he brings up CRISPR, and it just went, ding. Because they were talking about this in their classes and thinking, how do we prepare all of our students to maybe go on and use CRISPR in their jobs, or live in a world where CRISPR is a reality, where gene therapy could be the way that they're fighting a disease in their future, or helping a family member navigate that process. So really big questions that our team is always grappling with, and I'm so impressed and lucky to get to work alongside them. So Shu-Yee Chen is here today as well. She's our AP Bio teacher and one of the team that's been working to get a lot of this equipment into our classrooms. So I wanted to talk first a little bit about our program here at North. So as you may know, bio is a capstone experience for us. We start with physics in 9th grade, and then we, I can get those. They don't. We do this every day in class. Chemistry is our sophomore year experience, so that way when students get to biology, they have a lot of foundational knowledge that allows them to engage really deeply in the newest side of biology. It's not just ecology and plants, it's now genetics and DNA, and to really understand everything that's happening in just the mural that you see up here that kids have made for us. You really need this deep understanding. So it's exciting that we have juniors who are ready for a really high level of content knowledge. And as you know, biology has changed dramatically. We're now looking a lot at our genes and what we can do to actually change our genes or learn more about them. You can go down to CVS and buy a $200 kit, take a swab from your cheek, send it off in the mail, and within 2 to 4 weeks, you get information about your genes. You can get the raw data about your genome. And then the question is, who else has that access? So lots of big questions that our team is trying to answer. And to do that well, we want to equip students with the skills that they need to do that. So one of our big, most important gateway tools is called a micropipette. So, or a micropipette. So this is a micropipette, and we're actually going to do the activity that your students do in their junior year when they learn about micropipettes. So to do that, I want to introduce you to a few different parts of our tool. So the first one is the tip on the bottom. This micropipette is reusable, which is great, and you don't need to clean it. You just get it serviced every year. But the tips at the end are disposable, so that way we don't have to worry about contaminating our tool. They attach at the bottom. We'll show you in just a second, and then you can release. The other important part of a pipette is that it has a button at the top, which is how you actually draw up teeny tiny amounts of liquid that contain things like DNA. And there are 2 pressure points on this pipette. So the first one allows you to get rid of, you can't even see my fingers going down, but this allows you to dispense a small amount of air and then draw up some amount of liquid, which I've programmed into my pipette on the side. Then when I want to dispense, I press down past the first pressure point and all the way through a second pressure point or a second stop, because water, whether you know it or not, is actually kind of a sticky molecule. I'm a chemist by training, so this is where my light bulbs go off. When we press down a little bit further, water that's sticking to the side will actually be pushed completely out of the pipette, and you can dispense exactly the amount that you're looking for. So this is kind of a tricky tool to use, which is why we spend almost a full day doing a lab on how to use a pipette. So that's what we're going to do right now. Your job is, with the pipette that you get, you're going to be drawing up small amounts of water and dispensing it onto a piece of waxed paper and trying to make 10 equal-sized dots of water using this tool.
I've never brought water unless I like pour this in.
Oh, you can scoop up here.
Oh, yeah, so that's when you're going to end
Cool. Okay, you got the gist of it.
And then the best, well, this one doesn't have it.
How are we doing? We ran out of water. Oh, you did? Okay.
So I saw a lot of different sized dots. And just like we tell students, don't worry about what your dots look like compared to someone else. Because you all had different settings, and you may have noticed we actually have different size pipettes. So today we're using 2 different sizes. You'll find a P20 and a P200 in your kit. You should have 2 of each at your table. So just take a quick look, make sure you've identified which one's which. P20 versus P200. They tell you often right on top of the button that you've been pressing. Yeah, so this one is yeah. So you'll notice we also have a few different styles of pipettes. Our goal with this fundraiser is to just get a single style that can be serviced by a single company, and that allows us to have just a single method of instruction for kids. It's also one that's really durable and that will hopefully last us 25 plus years because we're getting them serviced regularly. So,
Heather, sorry, if I understand from our discussion before, I think the goal is to get enough micropipettes so that pairs of students could each have a touch on them. So it's not like we're trying to get one for each kid, it's just so that they're, you don't have one person sitting watching in a table of 6 or 5 or 10 or whatever it is, watching somebody, because it's a touch, it's a touch skill.
So we're going to do one more activity, and this time we're going to be learning about how to change the volume that's in a micropipette and how to check your work afterwards. So in a P20, what the red digit is telling you is after the decimal place. So if you see on a P20 015, this is actually 1.5 microliters, or 1,000th of a milliliter. 1.5 thousand, yeah, of a milliliter. And then on a P200, what this is telling you is the number of microliters. So if you see 020, this is 20 microliters. We're not using our P1000s today because those dispense up to a milliliter, which is not actually, in the scheme of things, a lot of liquid, but in our scheme of things today, and in the scheme of DNA, that's a ton of liquid. So we've set those aside. And we're just using these 2 today. Your next activity is asking you to make mixtures of colored liquid that match the standards that are in the center of your tray. So you'll see there's numbers 1, 2, 3, 4, 5, and 6. If you pick them up, you'll notice different colors, not our primary colors, and different volumes of liquid. So one of our biology teachers, Dr. Gibson, and her TAs have actually set up these standards for you, as well as little microfiche tubes full of red, yellow, green, and blue dye. And your job is to create another set of mixtures that you can check against the standards. So what you have here is a table that tells you what should go into each test tube. Okay. How much red dye, blue dye, yellow, green, and water. Okay. So that you can make these tubes. There's enough for you to make 2 sets at your table, but it's up to you. If you just want to work together to make 1 set, that's all right as well. So we'll take a few minutes to see what you can do.
If we get through one before the time's up, that'll be amazing.
4 and 5. It's the same.
Oh, but
Oh no, one's 10 and one's 2.
we're almost ready.
All of it.
But it's not. It's sticky, as you might.
It is sticky, right? So I have a few guesses as to why this is happening. Sometimes, even though if you draw up the code too quickly, like so when I press down to get an arrow, it does. And then if I drop really fast, sometimes you can get air bubbles. So if you want to drop kind of slowly, it's like a gentle release.
It's supposed to be like good.
So by doing this activity, I hope you feel like you learned a little something about pipettes. And that it's a little bit trickier than it seems. It's not quite as easy as just pressing down and lifting up. I don't know if some tables caught this more than others, but this also tests to make sure that you have every little part of the skill down. So if you have too much volume compared to the standard, it's very easy to see. If you're getting a lot of one color, like you can sort of identify which person in the group might be needing a little help with their pipetting technique, which is great as a teacher to be able to help target which students need what help. We also got to, Shu-Yee and I got to share a few of our tips as not quite master pipetters, right? Like that water is sticky, you can take advantage of that as you're letting go to do a slow draw up and down rather than a really quick one to get air bubbles. A lot of things to talk about. So pipetting is kind of this gateway skill to the rest of biotechnology. And I'm actually going to invite Shu-Yee up to talk a little bit about the things that she's done in her AP Bio class. We're just going to skip those for now. And what she does after students learn how to pipette.
So that was my original title. I changed it to Made It Formal, but after this I thought I would be fitting. It's not simple, yeah? We know that, okay, we're going to teach genetics, we're going to teach about what's coming up in terms of the new trend in STEM education and all the jobs that's around us, around Boston area, so many biotech companies. And I got some equipment through a grant and I tried some activities. Working with students, working with people is never as easy, but there's some fun stuff.
They're on different sets.
Yeah.
Yeah. It just doesn't matter.
Alright, so just a little context, I teach AP Biology. It's a 2nd-year biology course, so every student in the school has to take 1 year biology, usually during junior year, but not necessarily. So 12th grade, they come to me, they're interested or somehow interested in sciences, doesn't know what to do, or they want to be doctors. Those are kind of the students, the span of students I get, usually 27 to 28 students per class, depending on the year. And last year, when I was in molecular genetics unit, I did a few different activities to kind of pull it together. We did a transformation lab where we made mutant bacteria to glow under UV light, pretty cool. And we did some review of different technologies that's current and past technology as well to kind of set the stage. So students made infographics about different technologies. And the fun thing last year that I got to try with my students was they got to swap a little bit of their own cheek cells. We extracted their DNA. We ran it through a tiny machine to amplify it called the PCR machine. Just make lots and lots of copy of DNA. And ran it on gel, and what the gel tells us is, do they have this very particular repeated sequence from a virus or past virus sequence? So that's all it tells us. And why? not only was it cool, what we did is we took that information, so we took the class data and the percentage and compared it to the North America data and then the globe data. And it's essentially what you do if you decided to do a 23andMe or Ancestry.com. It's in a much bigger scale, but we did it in a smaller scale, a smaller setting. If you work in the biotechnology or biology or worked in the lab before, you know this machine. That was the old PCR, very chunky machine, and you have to program it. It takes a lot of steps, and students will load pretty much the whole class, or half the class will load their sample, and the teacher does the work. What was really exciting to me as a student and a teacher, when I went to the workshop and saw this machine, I said, this is amazing. It's a PCR machine that can make lots and lots of copies of your DNA. Just have it attached using a little tiny fan that's inside. And you put the tiny tubes in there, and after 3 hours, you get a sample of 8 DNAs that's amplified. Quite amazing. And what's cool, I saw this Edutopia video where these students go around to fish markets and supermarkets and grab samples of fish. Because there's a lot of rumors on the street and around the neighborhood saying the fish that they're selling you is actually not the fish. So it happens in restaurants with supplier issues. It happens in sushi places a lot. So students actually went and sampled different fish DNAs and amplified them and tried to kind of figure out what's going on with how people market fish. This is so cool. This is what I did last year. I'm not going to go into a lot of kind of details there. What was my main goal as a teacher? So I want them to think about the different applications of genetics. Not just, here's our DNA, it's inside us. What does that mean? What can we do with it now in this day and age? So they spend a lot of time researching about different things like DNA fingerprinting, from forensics perspective, to things like we look at how ecological systems can use genetic data to even think about that, like think about patterns, think about what's happening to different species. Then, for the actual lab part, they got to do cool things like they took their own DNA, they amplify it, they visualize it on the gel, and they took that information and analyzed the data. So, they say this was cool, and I learned a lot as a teacher every time I do something new, which was to keep it simple. The activity that you just did took us 2 days in the classroom. I did it a little differently. They actually got to make patterns that make this as NNHS, which they think is pretty cool. Pretty cool. But it took a long time. They need a lot of visual aids, so I spent a lot of time trying to think about what can I do to provide them that tools and all that. And I need to slow down a lot in order for them to have successful gel. You can't really see it under this slide, but they're actually, these are the student results that either shows if they're homozygous, if they have 2 copies of, if they have the repeat, if they are heterozygous, both copies have, 1 copy has the repeat, 1 copy doesn't, or if they don't have the repeat. So that's kind of what they got out of it. One thing I do want to think about, and as I work with my colleagues, one thing is to think about trying to make connections throughout the entire biology and the entire genetics unit. So this is not something that kind of just happens because someone likes genetics, likes biology. This is something that they have to make decisions about this throughout their whole life. This was my class last year, and the students were staring at their spit. They were staring at the spit, that's their DNA, and they were very, very excited. One thing about that is it's a 4-day lab. So if they screw up just a tiny, tiny bit for the day before the PCR, they have to put in 2 microliters or 1 microliters of reagents into these even smaller tubes than the ones you're holding. And unfortunately, if they didn't get enough practice, they don't get any results. Which is really sad after 4 days. We got pretty good results.
Why were they wearing the mask?
Why weren't they wearing the mask, or why were they wearing?
Yeah, why?
They were wearing goggles because it's just a standard precaution, even though it is their own DNA and their own spit. But it's just a standard precaution. There's nothing, the technology has changed so much that none of these are, it used to be some of the gel things are carcinogenic, and now we've done way beyond that. So this is kind of my journey as a teacher. The first year I took over AP biology, we did none of these. We had pipettes. We probably use them once a year in the classroom if a teacher has the time and the management to deal with all the equipment in the prep time. And I took 24 students to Harvard. They had one field trip workshop day where we got in. So we took 24 students and they tried this experiment, similar experiment. And then the next year, my colleague and I really wanted to push it, so we're still trying to push it. And we're hoping, the goal is to get every single student who are taking biology to have exposure. A lot of other schools does it a little differently. What they do is they offer a biotechnology elective. As a team, we really believe that we want every student to have this experience. Not only is it important, but it's also an equity issue, but also I think it opens a lot more doors for some of our population of students. I think this is the last thing I want to talk about, which is even if students don't care. So there are lots of colleges now that have biotechnology programs, so it's definitely a career path, and there are infographics and there are information out there on how important, how fruitful it is to be in kind of a STEM fields. Even if they don't care about that, going back to kind of where Henry started, this is a societal issue now, really. We talk about at dinner table. Do you want to put your DNA in that database? What information does that give you? Now this company offers information about your health. What does that mean? What does that number probability really tell you? So I think this is really where we want to think about as teachers and students to discuss, what do you want to do with this information? How easy it is to get, how accurate it is, and what are some choices you're going to have to make? Those are my students, and I do want to thank PTSO and parent community a lot because I've written many grants in the last 4 years to get to this place. Thank you.
So now is when I really want to make our case, because the pipettes that you've used today represent the majority of our collection here at Newton North. Every biology class that we run is somewhere between, for our CP classes, 12 students, to our honors bio classes and AP bio classes have 28 students. There are 3 different sizes of pipettes, and we need class sets of each size, not just 15 pipettes of random amounts. So this is a really big push for us because we want every student to be able to access this equipment. And we also don't want to run into this problem of we have 1 class set and there are 22 sections of 11th grade biology, in addition to 10 sections of biology electives available to seniors between AP Bio, forensics, marine biology, sustainability. So imagine that you want to use the pipettes for a week in your class because you're doing this PCR experiment. All of a sudden you reserve them for 4 blocks out of a week, and there's 22 other classes competing for these pipettes at the same time. We're a great bunch, very collegial, very collaborative, but I'm sure you can imagine this means that teachers are like pitted against one another to do these experiments in their classes, and we often end up asking ourselves, well, who gets them this block? How do we decide what's fair? And really what's fair is every student in every class. The other thing that we've run into is teachers are so eager to do these experiments that they say, don't worry about it. Shu-Yee, you can have the pipettes this week and I'll take them next month. When I'm in a different unit, I'll come back to genetics to do these experiments so that way my kids get access, but then it's not matching up with the learning that they're doing in class. So our teachers are super creative. We've been doing a lot with the very little that we're working with right now, but this has been something they've asked for many years, and folks on the PTSO can tell you that every year Shu-Yee writes grants up to the maximum amount that you guys give. Many of our pipettes our pipette men here, and also our mini-PCR machines, which is how we amplify DNA, as Shu-Yee mentioned, have come directly from the PTSO. We're so grateful, and yet we still need so much more. We now have class sets of everything, but we would like 3 class sets so that we can run them at the same time. The PCR machines especially are really tricky because it takes 3 to 4 hours to run. So if I have a class A block and we start PCR, now no B block class and no C block class can end up using these machines that day, and maybe a D block class can use them. So not only does it take like a full day per class, but that results in us needing quite a few machines. But we ultimately believe this is really, really important for every kid to get access. So before we share a video from the folks who started miniPCR, I want to share my story. So I went to a traditional public high school in right outside Chicago, where I grew up, and then for my sophomore year, I headed off to a math and science magnet public boarding school, where they took kids that really loved STEM and put them all together, and the state paid for us to have this education where we had access to all kinds of incredible tools. We had our own biotech lab. I got to do tissue culture as a sophomore in high school, if you're in bio. That's pretty incredible. We had all kinds of equipment available, and every Wednesday we had off of class so that way we could do science experiments and be scientists. And I can say that heading off to college, I knew I wanted to be a scientist. I thought I wanted to be a biologist, and then I became a chemist, but that's a different story. And it was because I had the confidence to use a lot of equipment that I saw in the labs. When I headed into my first college lab freshman year, and I had to pick up a pipette, it didn't make me flinch. It made me say, oh, I know exactly what I'm doing. Let me get to work, and let me start thinking about whose lab I want to do research in, which then got me to the next experience and the next experience after that. And I want every single one of our kids to walk out of here with that confidence. Even if they're not going to go on and do a degree in chemistry, if they go on to do an associate's in a health career and that gets them a job that doesn't require a college degree, that will pay a decent amount so that they can live in the Boston area, that makes me feel really good as an educator too. So the fact that we're training all kids for all kinds of careers is great. So with that, we were supposed to have Zeke, the head of miniPCR, come join us today, and at the last minute this morning he had to cancel, which we're really sad. He's okay, but in lieu of that, we have a video showing you what he and his partner have done in order to, as they say, democratize DNA. So.
Zeke and I grew up on the same street in Buenos Aires, Argentina, and we went to university together. We struck a good friendship, and then Zeke transferred over to Stanford. I stayed, I got my degree in Buenos Aires, and then was lucky enough to come to Harvard for grad school. And then I think it was my second day in the United States, I'm walking on the quad at Harvard Medical School, and I bumped into Zeke and his wife. We both had this passion that genetics and DNA analysis should be more accessible. How do we actually do DNA analysis where it's needed but not available today? Zeke and I were both passionate about the same problem, and at one point decided, let's just take the leap and do something about it. And we founded miniPCR.
What we have done is we've taken this technology that is normally very large, it's very expensive, it's difficult to use. What we've done is we've made that technology a lot simpler and a lot smaller and a lot more affordable.
The traditional PCR machine that I used in my PhD is more like the size of a big printer. It's a big problem if you're a researcher out in Africa trying to diagnose Ebola. If you're a student in a classroom like I was, who's just curious about DNA and wants to know what it's all about. Zeke had this idea that it's not rocket science. We can make this a lot better, a lot cheaper, a lot easier.
And we came up with this very small machine that can work just as well as machines that cost $8,000 or $10,000.
We were contacted by Boeing and Cases, who had been looking to fly PCR technology to the International Space Station.
Genes in Space is a competition for students in grades 7 through 12 to think about an experiment regarding DNA to be sent to the International Space Station.
Liftoff of the Falcon 9 rocket.
This could be a fascinating project for kids to engage on, to actually do pioneering DNA science in space.
miniPCR is a tool that is accessible for all. So the same technology is being used for forensics. The same technology can be used in classrooms, and students can use it the same way that you would use it in a lab. So it is just a tool, and it is very powerful and very sophisticated. This is a tool that enables you to indulge in your curiosity.
This can really change education. This can really enable kids to actually explore their passion for DNA science early on.
We want to change people's relationship with biology. We want them to have a direct connection.
If you have any, and then Alexia, Sally.
So how much are the mini PCRs?
They're $650 a piece. And right now we have one cross set. For multiple.
And a class that's not one per person, it's one per group, right?
Yeah, so it's 7 mini PCRs.
How much are the reagents for each time you do the PCR?
So that's a good question. They sell kits, and we're able to keep it under $200 per class when we do mini PCR, and we
For
can
the whole
actually,
experiment?
yeah, and we can handle that within our annual budget that we get from Newton Public Schools, because the team is really committed to doing this.
And there are cheaper kits if we're not talking about extracting DNAs from tissues. So, for example, I've worked with students where we just look at, so we can get sample DNAs and they can amplify it. So it's cleaner, it's easier, it's less steps. So kind of it's definitely a place that we would think about for differentiation to help students to kind of start that first step.
Do you have most of the other equipment you need as well, like the microtubules and electrophoresis and everything else?
Yeah, we have more gel boxes than we will ever need to use at one time. Yeah, so we're lucky that we have the centrifuge, yep.
You have the freezers and everything else as well?
We do, yeah.
Minus 20?
Yep. So at this point, we just need pipettes and thermocycling.
What does PCR stand for?
Great question. I did prepare slides on that. Wasn't sure how much we were going to want to go into it, but since you asked. PCR is a polymerase chain reaction, and if I can back up. It allows us to amplify or make many copies of DNA. So when you extract DNA from something, you end up with really, really small amounts. And then we typically end up looking at one part of one gene, and we just want to take a look at what that sequence is. But before we can do anything with it, we have to make enough copies that we can read it, sort of like making it brighter. And so what PCR does is it
polymerase chain reaction.
So this is DNA. I'm going to hit the lights for just a second. What a PCR machine does is it heats up a sample to a really high temperature, and then DNA breaks apart at that high temperature. It then brings the temperature back down, and then you get basically the start of a photocopy happening, where more DNA sort of attaches to these separated strands. Then we heat the DNA back up just a little bit, not all the way. It's going to do that in just a second. And then an enzyme can attach to DNA and make a complete copy of the sequence that we're interested in, which is going to do in just a second. Yep. And now, all of a sudden, instead of just having 2 copies of something, we have 4. And then we do this process all over again. So the next time we heat up, we end up with 8 strands. The next time we go through the cycle, we have 16. So you end up doing somewhere around 30 rounds of something like this, where you heat up, cool down, heat up a little bit, cool down, heat up a lot, etc., so that you can end up with about 1 billion copies, which is enough for you to be able to read on a gel that Shu-Yee showed you, so that you can actually take a look at what the gene sequence is. So
just to go into kind of a little pedagogy, why we think this is really cool is, in terms of DNA replication, how our cell naturally copies DNA, it's mimicked in a machine, but this is when students actually see this, when students actually hear the cycle goes up and down, and when they can see it on the computer and seeing the temperature changes, we start talking about enzymes, we start talking about how DNA replication actually works, and how do we take that in a biotechnology way, and that's when they start really understand what they just read in the book. Why do we need polymerase? Why do we need primers? And that's when they really make sense to them.
And this particular machine plugs into a computer via USB, and you actually get a graph of all the temperature changes that are happening. And kids can do calculations as that happens, like how many sets of DNA do we have now? And like 30 minutes later, how much DNA do we have? And they can also change all those temperatures. So if they wanted to go in and do an inquiry experiment when they get to AP Bio and they have this year-long inquiry project that they do, they could potentially use these machines to figure out how to get the best copies of. So thanks for asking. Anything else?
So to fit this in the curriculum, what did you have to give up?
I talk faster. So that's one thing that's, as I said, it's very much a learning experience. So all I did last year was saying I would just spend a little less time here and there and catch up. There are definitely much more better ways of incorporating each pieces when I teach different pieces. So for example, like I now teach DNA replication with talking about PCR machine. So they really understand what they're doing. And I now start talking about, when we talk about enzymes, we talk about why that 72 degrees is so important. We need to find an enzyme that can work at that temperature. So it's kind of it does take some time and planning and some failing to really learn about when is appropriate. And really the cool thing, that's why I'm a biologist and I teach biology, is that you can really incorporate it throughout the whole year. It's something that's universal, and with the technology, with the news, with the internet access, students can learn about all different components throughout any unit and you can connect it together. So.
And I hear from our CP to honors bio teachers, I think we do a little bit less ecology than we might have 10 years ago, but we're also evolving with the standards. The master curriculum frameworks changed in 2016 for science, and there's a bigger emphasis on genetics and molecular biology, so we're also pushing to meet those.
Are any of the other sciences taking these leaps forward that you're trying to keep up with? Or?
Yeah, I mean, all of our science courses are trying to keep up with the new curriculum frameworks. Physics is where we take the MCAS, so we're sort of held more accountable by the state for that curriculum, and yeah, our curriculum's going to be changing quite a bit. And part of it is that the middle school is taking over a lot of what we used to teach. They do motion and Newton's laws in 8th grade now. So we'll be able to do things like electric fields and nuclear chemistry and wave particle duality, which is now part of our 9th grade curriculum.
So thank you so much for explaining this. One of the things that we thought would be fun to do for those who wanted was one of the things that we've heard that Melissa and I learned that they do in biology is they take this test to see if they have the gene that allows them to taste this PTC, which is a chemical, an enzyme, on these papers. So we did this beforehand, and it's sort of part of their genetics unit, and a group of us just did it beforehand to determine whether we have the gene to taste it or not. And I can tell you that Principal Turner has the gene because apparently if you taste it, it's very bitter. So we have candies as well. And Melissa and I are not tasters, so that turns out that that's actually more rare than being a taster. So if any of you are interested, I have these little strips. The kids are doing this, so it's kind of fun, if you have a senior or a junior, or even an upcoming junior at some point, to be able to let them know whether you have the gene or not, because they'll be doing it as well. Isn't that correct?
Yeah, and so the thing that we didn't say, which I apologize for, is that one of the PCR labs that we can run is actually to amplify the sequence of a student's genome that tells them whether or not they are a taster. So they get the experience of saying, oh, I really taste this bitter taste, that's disgusting, or what's going on here? And then they run their own DNA on a gel after amplifying it, and they can see, oh, my sequence set is different than someone else's. So
So that's kind of fun because they've got the visual and they've got the actual sort of taste test portion of it at the same time, so.
So the way I explain in my classroom is, that's like when we go to the doctor, we say we have all these symptoms. He's like, well, I'm gonna do these things. Now we go to the doctor, I'm gonna take a drop of your blood. I can know exactly if you have these pieces. So it's kind of like the two different ways of looking at it.
Exactly, experiential. So anyway, this is an opportunity that's open to you. We all did it before, and we want to thank you so much for coming. Elizabeth, did you want to make your remarks or?
No,
just
thank you very much for being here.
And
And
if you have any questions, Melissa and I are around. We're available by email. We're excited to be running this campaign. We hope that you will support the campaign, and we also hope that you will talk to the people in your lives, including your students, about what you learned tonight, but also about what this year's campaign is about. Because as I mentioned, we do have a really ambitious goal, but as I think you can see, it's actually to bring some real life and awareness into this program so that everybody has access for it to these tools at Newton North and to the learning. So, thank you very much.
is there a time component to the goals?
Yeah, so our hope with this, obviously, thank you for asking that. The question was, there a time component to it? Just to emphasize that the sooner we get the money, the sooner the tools actually go into the classroom. So if you have a junior, we are looking at getting these tools into their hands this year. In fact, I think I mentioned earlier that we were talking before you all arrived about getting the bids in and making the first purchase order now.
Before we leave for a break.
Yeah, so we really are, as soon as the money comes in, we're going to get it out and into the classrooms. So yes, there is a time pressure on that. Yes.
And you'd prefer us to donate online or write checks, or what do you,
We have no preference.
Either
We
way.
have no preference. We have no preference at all.